So instead we'll talk about seeing color, which starts on page 310 in your book. We likely evolved to see color so that we could spot fruit. If you remember that we were initially hunter-gatherers, and being able to spot red fruit was very important to our survival. The primary colors of light, or the basic primary colors, at least for the trichromatic theory, are red, blue, and green. So note that this differs from the primary colors in like paint, which is red, blue, and yellow. Trichromatic theory accounts for very early color processing very early in our visual system. And it's an explanation of color vision based on the coding of 3 primary colors of light. And this theory says that the color we see, any color we see, is a function of the relative number of red, blue, or green sensitive cones on our retina being activated. And it predicts that if someone's missing one type of cone, the number of colors they see is going to be restricted. So if they just don't have red cones, they're not going to see the colors that involve red. And this is true. This is the case in color blindness. So a little more detail about color blindness, that you have some context. Lacking red cones and lacking green cones produces similar results. As you can see down at the bottom, an individual who lacks red cones or green cones is going to see a similar picture as compared to somebody who has all 3 types of cones, as in the first picture. But red-green is the most common type of color blindness. Red or green cones that are lacking can be either functionally limited or completely lacking. So there's different sort of levels of color blindness. There's a rather, I was actually surprised to see this when I read a bit more about it, that color blindness has a prevalence up to 8% in men and 0.5% in females. So remember that it's X-linked, so unfortunately men are disproportionately affected by color blindness. If you if you wonder if you have color blindness, if you aren't sure, apparently some people find out relatively later in life. There's this test that you can take. And there's also these kind of cute videos of people using glasses that fix color blindness, seeing full color for the first time, that you can see on YouTube. As well, many animals, such as dogs and cats, lack the green cones. It helps them actually see camouflaged items because they can't see these, because they don't pick up on green so much. And humans lacking green cones can actually see through color camouflage. Camouflage is not as effective for them. So sometimes the military uses humans who lack green cones for this reason. So the trichromatic model that we were just talking about explains the very first steps of color processing, and later processing of color can be explained by the opponent processes theory, which assumes that there are 3 pairs of opposing colors: black, white, red, green, and blue, yellow. And this is based on the behavior of retinal ganglion cells. So just a little introduction to this. On the next page, you can stare at the red-blue box for 30 seconds. And after you do that, focus on the white box next to it, and you should see a green-yellow after image on that white box. So if you want to try this, go ahead and pause your video and stare at the colored box for 30 seconds, and then stare at the white box, but I'm going to move on. Oh yeah, and this is another one. This is a famous individual that you might not be able to recognize based on this negative image, but for similar reasons as the box before, if you stare at her image for 30 seconds as well, and then stare at the white box, you should be able to recognize her. Okay, so back to the opponent processes theory. Retinal ganglion cells respond differently to stimuli in the center of the receptive field as compared to the outer portion, and it's actually quite an opposite reaction that they have in the center versus the outer portion. So their kind of baseline response is this like intermittent firing, and this is a response to white light. In the case of a red-green sensitive ganglion cell, the cell, I'm kind of anthropomorphizing or personifying the cell here to explain it, but it sort of like prefers red in its center. And since green is the opposite of red, it likes green in the outer circle, so it kind of wants the opposite. The presence of green on the outer circle is almost like having red in the middle. It kind of gives the same strong response. When there's green light in the middle circle, it doesn't like this, and it gives a weak response, but it's still a weak response that's a little bit less than its baseline response. When there's red in the center and green on the outside, this is its absolute favorite, and it's a very, very strong response. There's strong contrast between these two colors. And then when there's green in the center and red on the outside, it does not like this, and there's no response. The cell sort of expresses its preference through the frequency of action potentials. So more action potentials for the color combinations that it prefers, and this is how these retinal ganglion cells code for what color they are seeing in the receptive field. So color constancy is this other function of color in our brain where we see colors when there are multiple colors in a system and we already know what they are, or in our visual field and we already know what they are, they will look similar even when there are changes in luminance in the environment. And the V4 in the cortex, so visual cortex, is likely responsible for this. So a while ago, maybe last year, there was this image of this dress, the middle image here, that was circulating, and people couldn't seem to decide whether it was a black and blue dress or whether it was a gold and white dress. And the reason that we were not able to distinguish this is because we didn't have context and weren't able to establish what the colors were and then maintain this even in different luminance. So it was kind of poor luminance that made it unclear what these colors were. And in case you're wondering, I think somebody did some research and figured out that the dress was actually blue and black, the blue and black one. And then up in the corner here, this is another luminance illusion. And so typically people would perceive the bar in the middle of the box to be starting out light colored and getting darker, when in reality the bar is all the same color. So to test this, you can just cover up the background and notice that the bar is actually all the same color of gray, but because the context is changing behind this bar, it makes it look like the bar of the actual color is changing. I'm sorry, the shade of the actual bar is changing. Okay, so section 9.5, the visual system in action. Injury to the visual system can show some pretty interesting effects. So injury to the pathway on the way to the visual cortex. If the retina or optic nerve is destroyed, so if something early in the visual system is destroyed, the sight is lost in that corresponding eye, and the sight is lost, of course, ipsilateral, because if it's before the optic chiasm. Lesions to the primary visual cortex can cause scotomas or blind spots in the visual field, and that would be in the ipsilateral visual field. So notice in this image, in the picture, the damage is to the left visual cortex, and the scotoma is in the right visual cortex. And the degree of scotoma is dependent on the degree of injury. So a bigger injury causes bigger blind spot in that visual field. Blind sight is a condition in which someone who has severe damage to both sides of the V1 might report having no sight at all. So they would say, they would report being completely blind and genuinely feel completely blind, but there may still be some visual information being processed that is not available to the conscious awareness. So someone with blind sight may be able to guess details of an object in front of them with above chance accuracy. So if you say, am I holding a picture of a dog or a cat in front of you, somebody with blind sight would be able to, they would feel like guessing to them, but they would be accurate above chance. So there would be some kind of visual processing happening. Similarly, if someone throws a ball at them, they might move their hand to catch it, sort of involuntarily. So if you throw it sort of at them like it's going to hit them, they might automatically move to catch it. They might not catch it, so the visual system is not good, but they would react to that visual information. So injury to the what, or ventral pathway, which goes through the temporal lobe, can result in a number of different things. One of them is visual form agnosia, or the inability to recognize objects. So agnosia in general means like lack of knowledge. A means without, nausea means knowledge. Visual form, agnosia means the inability to recognize visual form. and there's some YouTube videos you can watch of patients who have visual form agnosia, but on the right is also an example of what somebody with visual form agnosia might draw in trying to replicate the drawing on the left. So really just not able to process visual form very well at all. Achromatopsia is the inability to distinguish color, and this is damage to the V4 region, so kind of the extrastriate cortex. Akinetopsia is the inability to perceive an object that's in motion. So individuals with akinetopsia can still see stationary objects. This is caused by damage to the V5 region. They can still see stationary objects, but once the object starts moving, they literally just can't see it. It's like the object disappeared. When the object is still again, they can see it again. And so patients with this damage have described that it's difficult to, for example, like pour water into a cup, because it just seems like the water is stationary and they can't see the glass filling up, so they might fill it to full. Akinetopsia can actually be temporarily induced by TMS, so transcranial magnetic stimulation. So you can put that coil, that figure 8 shaped coil, over somebody's V5 cortex and cause a temporary inability to perceive an object in motion. Injury to the how or dorsal pathway can result in orientation agnosia, which is this inability to recognize or inability to orient objects correctly. So there's a task where an individual has to rotate a card such that you can put it into a slot at the correct orientation. And you can see these circles with the lines through them down here at the bottom is like a control patient. The slot in this case was straight up and down in that task. So control patient, for the most part, was always able to insert the card at the correct orientation into the slot, whereas the patient, if they just had to match the orientation, like if they didn't sort of have feedback from the slot, they were almost completely at chance. They were unable to move this card in the correct orientation into the slot. In hemispatial neglect, an individual, this is damage again to the dorsal pathway, and in this case the individual doesn't process or neglects one side of the visual field due to this lesion in the contralateral dorsal stream. So if they have a lesion in the right dorsal stream, they're going to neglect the left side of their visual field. And this clock drawing is an example of a drawing you might see if you ask somebody with hemispatial neglect to replicate a clock. So you show them a picture of a clock and ask them to draw this. They know that there's 1 to 12, but they literally just don't process, or it really seems like they almost just ignore the left visual field, or the left half of the picture altogether. There's another example of a patient with hemispatial neglect who was asked to recall, I think she lived in Rome, and she was asked to recall like a famous square in Rome and describe everything on this famous square. There were many sculptures and things like this, and she closed her eyes and described everything she could sort of visualize in the square, and when they sort of drew it out, they realized she was only describing things that were on the right side, the side that didn't have hemispatial neglect. So she just didn't even have visual memory of the left side of her visual field. Okay, and this is just a quick summary that the ventral visual pathways are kind of associated with recognition, and the parietal visual pathways, or this dorsal visual pathway, is associated with the how, or like grasping objects using the correct orientation of your hand, etc., things like this. Okay, and I believe that's it.